Hello, dear students. Today we are going to discuss an important topic of molecular biology, that is central dogma of molecular biology. This is an important topic with respect to the board exams and also for the competitive exams, especially for the NEET examinations. In molecular biology, you will see that there are three important molecules, also known as icons of molecular biology. And these three molecules are: number 1, DNA; number 2, RNA; and number 3, proteins. Three important molecules of molecular biology. DNA is known as deoxyribose nucleic acid. RNA is known as ribose nucleic acid. Proteins, as we know, are the important biomolecules. Now, what we are going to study in this lecture is that what does central dogma of molecular biology means. We know that in living organisms, in all the living organisms, and also in some viruses, that means in all the living organisms and also in some viruses, it is the DNA which acts as a genetic material. That is, DNA acts as a genetic material in all the living organisms and also in some of the viruses. What does it mean? That means it is the DNA which has the information. It is the DNA which carries the information from parent cells to daughter cells. It is the DNA which carries the information from parents to their offsprings. And DNA controls all the characters of an organism. Now, what happens in central dogma of molecular biology? If DNA has an information, then this DNA gives information to RNA. That means if DNA is the master molecule, it has its messenger, that is RNA acts as a messenger of DNA. Then RNA carries this information for the synthesis of proteins. You will see that this information flows in a unidirectional manner. That means from DNA to RNA and from RNA to proteins. You will see never the information being carried from proteins to RNA. This is not possible in molecular biology. From proteins to proteins, you will see it is not possible in molecular biology. This information, that is the transfer of information from DNA to RNA, then from RNA to proteins, it is a unidirectional flow of information. And this unidirectional flow of information is known as central dogma of molecular biology. Another process is added here, If DNA is the genetic material, DNA also synthesizes its own copy by a process known as REP, replication. By a process of replication, DNA synthesizes its own copies, new copies, and from DNA by a process known as transcription, RNA is synthesized, and from RNA, the information is carried for the synthesis of proteins by a process known as translation. Then you will see that there are three important processes. That means if synthesis of DNA from DNA, that is this first process, it is known as replication. Synthesis of RNA from DNA, it is known as transcription. That is this process, second process. And synthesis of proteins from RNA. Synthesis of proteins from RNA, that is third process, it is known as translation. These are the three important processes involved in the unidirectional flow of information from DNA to RNA and then from RNA to proteins. It has been stated by Francis CRICK in 1958, also known as prophet of molecular biology. The term also has been coined by this Francis Crick, and also this process of molecular biology has been also stated by the Francis Crick. What he stated that in molecular biology, you will see if DNA carries the information for the formation of organism, for the formation of proteins, for the formation of particular molecule, that DNA carries that information to the RNA. That means this RNA acts as a messenger to DNA. Then DNA gives that information for the synthesis of proteins by a process known as translation. And this information never goes in a reverse direction. That means from proteins you will never see that RNA is synthesized, and from RNA DNA is synthesized. Though this central dogma of molecular biology has slightly got modified after the discovery of an enzyme known as reverse transcriptase in retroviruses by Temin and Baltimore. This enzyme is very important enzyme in these viruses, retroviruses. For example, there are some examples of retroviruses like HIV virus. It is a retrovirus having a reverse transcriptase, where you will see that single-stranded RNA acts as a genetic material. Then from this single-stranded RNA you will see by a process known as reverse transcription, single-stranded DNA is synthesized. Single-stranded DNA is synthesized, as this information is a reverse of the normal process, that is transcription, it is known as reverse transcription, reverse of normal transcription. From single-stranded DNA, then a complementary strand of DNA is synthesized, then this single-stranded DNA is converted into the double-stranded DNA. By this, the process got slightly modified, and we can also write that in the normal process. If normal process is like this, I will show here. If this is DNA, from DNA you will see DNA is synthesized by the process number first, that is replication. Then another process important known as transcription. By transcription, RNA is synthesized, and a reverse of this, by the reverse of this process, that is the reverse transcription. Reverse transcription, DNA acts as a template, and upon this DNA and RNA acts as a template, RNA acts as a template, and upon this RNA, DNA is synthesized. That means this is a process which occurs in retroviruses like HIV virus is having an enzyme reverse transcriptase, and this central dogma of molecular biology got slightly modified. Then from RNA you will see proteins are synthesized. This is the modified version of the central dogma of molecular biology, while you will see that a reverse transcription has been added, and this reverse transcription is also known as a process known as tamanism, as the enzyme reverse transcriptase has been discovered by this scientist. Thank you for this video.